Chapter 2 Orgone Energy Vesicles, Bions and the Natural Organization of Protozoa. Experimental foundation for understanding the cancer biopathy. Orgone energy was discovered in a bion culture. My first task, therefore, is to give an account of the orgonotic phenomena that represent transitional stages of evolution between living and non-living matter. Because of the functional relationship between bions and atmospheric orgone energy, it is essential that a discussion of orgone functions in bionous matter precede the presentation of the actual discovery of the orgone. It is difficult to determine the date for the discovery of orgone energy. Orgonotic functions of attraction, penetration, pulsation, and lernation had already been observed in the period between 1936 and 1939, and had been subjected to investigation in a variety of bion preparations. However, I had no presentiment that I was working with manifestations of a specific biological energy. Experiments with bion cultures led to the discovery of orgone energy in SOPA, San Packet, Bions during January 1939 and in the atmosphere during July 1940. It was only after I had worked on the purely physical functions of orgone energy, 1939 to 1942, that understood the observations I had been making on bions and bion cultures since 1936. The description in my book Die Bion, 1938, conforms completely to traditional bacteriological and biological concepts. 14 Orgone energy vesicles, bions. 15 Later knowledge of orgone functions caused me to modify much of what I had written in that book. For instance, cultures of cocci and bacilli derived from bions represent not, as I thought then, a more advanced stage in the development of the bion but, on the contrary, a degeneration of bions to a biologically sterile form incapable of further development. I found that bions actually develop in the direction of protozoal organization. On the other hand, staphylococci, streptococci, T. bacilli, and rot bacteria are due to a degeneration of the orgonotic living plasma. Mistakes, such as the one I just mentioned, and the subsequent necessary modifications are unavoidable in working in unexplored territory. The following account of the bion experiments is given in the context of knowledge of atmospheric orgone energy, and its perspective is therefore no longer biologistic but functional, based on energy principles. Bion and energy vesicle designate one and the same microscopically visible, functioning formation. The term bion refers to the vesicles into which all matter disintegrates if made to swell. These vesicles represent transitional forms between non-living and living matter. The bion is the elemental functioning unit of all living matter. At the same time, it is the bearer of a quantum of orgone energy and, as such, functions in a specifically biological way. It is an energy unit, compounded of a membrane, a fluid content, and an amount of orgone energy, that is, an orgone energy vesicle. In what follows, I would like to give an account of the observations and experiments on which the far-reaching conclusions just summarized are based. 1. The vesicular disintegration of swelling matter. Palmon's carbon is the fundamental substance that, when combined with oxygen, nitrogen, hydrogen, and water, forms the basis of the orgone energy vesicles, bions. 17 Which is motionless. In the dark field, we observe an essentially striated structure interspersed with occasional vesicular formations, along the margins, between the striated structure and the tiny oval-shaped vesicles. The light is strongly refracted. We set the microscope at a magnification of approximately 2000x, objective 80x, IP16x, and inclined binocular tube, which increases magnification by 50%. The striated, vesicular structure can now be seen more clearly. There is not the slightest indication of movement. We add a drop of ordinary water and look again, first at 300x then at 2040x. Essentially, there is no change. We see no movement, only here and there round or uneven particle may be in motion. Its size seldom exceeds 1 micron in diameter. Overall, the field is lifeless. There is no sign of contraction or expansion. Using a spatula, we add finely pulverized carbon powder to water contained in a test tube. A portion of the powder sinks to the bottom, while the remainder floats on the surface. The water itself retains its clarity and no colloidal solution forms. The preparation is not sterile. Each day we draw off a drop of the fluid and examine it under the microscope. The object is to discover whether changes are taking place in the carbon, and if so, what kind. But a change is observable only after several weeks. The tiny individual spherical particles moving weakly across the field are becoming more numerous. In the dark field, the larger particles of carbon display a very gradual increase in the number of spherical formations within them. However, the overall scene remains unalive. Months pass without much change. We are struck by the absence of ordinary air bacteria. The test tubes are of course sealed with cotton plugs. Macroscopically, the fluid appears unchanged. It is still clear. This is our control experiment. The experiment for the production of coal bions is as follows. From now on, we apply strictly sterile procedures. All liquids are autoclaved at 120 degrees Celsius. All dry substances and instruments are dry sterilized at 180 degrees Celsius. Test tubes containing a preparation of 50% bouillon and 50% 16 Wilhelm Reich variety of organic compounds as well as living matter. Our intention is to ignore the already well-known chemical reactions and simply concentrate on examining a particle of carbon under a good microscope equipped with epichromatic lenses. Reichert microscopes, the Z microscope, which permit magnification up to 5000x, were used for the experiments. The finer biophysical processes like expansion and contraction, vibration, and formation of a radiating bridge can be observed only at magnifications greater than 2000x, and are best seen at 4000x. What matters is not the resolution of fine structural details but rather the visualization of movement. For this purpose, we can use carbon, derived from blood charcoal, obtained from Merck & Co., or from ordinary soot. Since the process of combustion converts all organic compounds to carbon, the origin of the carbon used for the experiment is not important. First, we examined a particle of carbon dry, at a magnification of approximately 300x. What we see is a black, uneven structure. 0.1 NKC1 solution are autoclaved. Then we heat a small amount of coal dust on a spatula tip in a gas flame to white incandescence. While the coal dust is still white hot, it is plunged into the sterile fluid. The fluid immediately turns black and only the heavy particles of coal sink to the bottom. The lighter particles remain suspended. A colloidal solution has been formed. In contrast to the control experiment, over the course of a half hour, the black fades to gray, The solution remains colloidal for 3 to 6 days, then it becomes clear. All the particles have sunk to the bottom. The preparation completed, we draw off a small drop, using sterile procedures, and examine it under the microscope in bright and dark field, again starting with a magnification of 300x, cf. Figure 25, Appendix. Then using 2000-3000x, for bions seen at this magnification, cf. Figure 30, Appendix. What we see is fundamentally different from what was observed in the control preparation. The structure of the individual coal particles is primarily vesicular. With continued observation, we are able to see small vesicles approximately 1 micron in diameter disengaging themselves from the margins of the larger particles and moving about freely in the fluid. When the preparation is successful, movement may be observed at the margins of the particles, expanding, contracting, vibrating, etc. But even the smaller particles that move about appear to change before our eyes if we observe long enough. First, they appear hard, the membrane black and thick. Gradually, however, the membrane becomes thinner. On the inside, we see increasingly a blue and blue-green glimmer. The vesicles become more taut and show increasing internal movement. Wave-like vibrations may be observed in many vesicles. The thinner the membrane becomes, the more intense the blue and the more elastic the movement. To underscore soon, on the same or, still better, on the following day, we can clearly see movements of expansion and contraction. No one who has studied these preparations for any length of time can doubt the living character of these movements. We distinguish movements of the vesicles from place to place and inner movements Orgone energy vesicles, Bions, 19 of their contents, fluctuations of the blue color, variations in brightness, protrusion and retraction. The vesicles pulsate with an irregular rhythm. Will pass a galvanic current of approximately 0.2,0.5 mA. Through the preparation, the vesicles move toward the cathode and therefore have a positive electric charge. After several days, when the particles are no longer in colloidal suspension, the cataphoretic phenomena fade or disappear altogether. The charge of the vesicles seems therefore to be a prerequisite for colloidal suspension and motility, as Pauli surmised. It is also a prerequisite for the capacity to form cultures, cf. Bion, pp. 54ff. We try an experiment with biological stains, using gram stain or carbofusin. As a control, we stain plain coal dust. The unprepared coal does not accept any biological stain. The particles remain black. The coal bions, on the other hand, show a positive stain reaction, blue when gram stain is used. It can also be observed that the staining is restricted to those particles that have attained a certain degree of bionist development, thin membrane, increased fluid, blue on the inside, while the undeveloped particles react neutrally, like those in the control preparation. We examine the stain preparation at a magnification of 3000x, using oil immersion, and find that most of the blue vesicles that previously had every possible form have now become spherical. A new phenomenon is especially striking. Alongside the large-sized vesicles, approximately 1 micron in diameter, there are tiny red bodies which were not visible at a magnification of 300x. The smallest of them are approximately 0.2 micron in length, that is, only barely visible microscopically. They lie in groups around the larger round, blue vesicles and unstained crystals. They are elongated, and are pointed at one end like miniature lancets. They were not observed in the fresh, wet preparation, but can be found in a live state in other cold bion preparations. Photograph of blood charcoal preparation at 5000x magnification, cf. Figure 26. Appendix. After long experimentation, it became clear that these gram-negative bodies are of the greatest significance. They are the so-called T bacilli, which play such a crucial role in cancer. More on this subject later. Our conclusion is that bions are biologically active. Worms cause, in contrast to the substance from which they originate, they react to biological stain. There is another specifically biological characteristic of bions. Non-living substances viewed under the fluorescent microscope always show only their own characteristic color: coal, black, sodium chloride, yellow, etc. Cold bions viewed fluoroscopically show not a black but a blue glimmer, as does a Staphylococcus culture or any organic cell tissue. This is additional proof of the biological character of the coal bions. Before proceeding to an investigation of other properties of the energy vesicles, we must establish whether the blue vesicles develop exclusively from carbon or from other substances as well. If they were to be found exclusively in carbon, the fundamental question concerning the nature of biological energy in non-living matter would be easy to answer. But the problem is complex, because the more substances we examine and subject to swelling, the more the following conclusion is confirmed: all matter heated to incandescence and made to swell consists of or disintegrates into blue glimmering vesicles. Cooked foodstuffs, muscle, when it is cooked, loses its predominantly striated structure and consists of blue, motile vesicles. The same results are obtained with every kind of vegetable. Size and shape of the vesicles may vary, but the content invariably shows a blue glimmer. Egg yolk consists of individual blue vesicles, sometimes in the form of a heap of vesicles surrounded by a membrane. Milk contains, apart from fat globules, blue bions. The same applies to cheese, especially those varieties processed with the aid of bacterial fermentation, for example, kefir, Roquefort, yogurt, and casein of every kind. Vitamins, examined at a magnification of 2000x, consist of blue vesicles, irregular in shape, which refract light strongly. Egg white when raw is without structure. When cooked, it disintegrates into heaps of blue vesicles. Blood serum reacts similarly, and blood platelets along with the red and white blood corpuscles show an intense blue glimmer. The structure of moss and grass is striated without vesicles, similar to that of animal muscle. When both are cooked, they disintegrate into blue vesicles, which look like algae. This raises the question whether the algae found in stagnant ponds are not the same as our bions, namely, matter disintegrated into energy vesicles. Blue vesicles abound in stagnant water, serving as foodstuff for protozoa. The bion experiments yield a surprising answer to this question, which we will discuss in a different context. We try to find out more about the formation of bions. We mix certain structureless substances together in a certain sequence. First, we make the following solutions: A. 100 cc. Water plus 50 cc. 0.1 NKCI plus 2 mg. Dissolved gelatin plus 50 cc. Filtered bouillon. B. A few drops of egg white in KCI. C. A small amount of fresh lecithin in KCl. These solutions show no structure. Only the lecithin mixture shows space enclosing membranes, without an inner structure. We now mix the three groups of substances. Within minutes, the blue bion vesicles are visible microscopically. Previously there had been no motion of any kind. But to underscore, now the solution is swarming with moving forms. The gelatin combines a number of blue vesicles together into a heap, which contracts and expands. The effect is one of individual vesicles inside the heap straining to move in a variety of directions and thereby generating inner motility. In general, 4 types of motion can be distinguished. If we now add finely pulverized blood charcoal, we can follow the development of highly motile coal bions. We witness the carbon absorbing the fluid containing egg white. The previously empty lecithin tubes fill with vesicles. The whole scene is one swarming with life. We then autoclave the mixture. The movements become even stronger. Now the T bacilli appear. The biological stain reaction, carbon fusin, Giemsa or gram, is now positive. In January 1937, I rendered art account of this experiment, number 6, to the Academy of Sciences in Paris. In January 1938, I received word from Professor Lapicque that, after 1 year, the autoclaved preparation still showed lifelike movement and contained contractile forms. The news was all the more surprising since the preparation was sterile and sealed, airtight. Here is Professor Lapicque's letter, translation of proceeding, University of Paris, Sorbonne, January 25, 1938. Faculty of Sciences Laboratories for General Physiology, 1, Rue Victor Cousin, 5. My dear Doctor, requested by the Academy to study your communication of January 8 of last year, I first waited for the arrival of the film you were to send. Then, since I did not receive it, I examined microscopically the samples you included with your initial communication. I have in fact verified the lifelike movements that you described. That fact itself is remarkable considering the length of time that has elapsed since the preparations were made. I should like to propose to the Academy the publication of your findings in brief, together with a short annotation by myself confirming the fact and offering a physical-chemical interpretation representing my own personal viewpoint. Would you agree to the publication of your contribution in the excerpted form attached, which is actually a resume of the important part, while leaving out your electrical theory, which has nothing to do with the experiment? It seems to me that this arrangement would be in accord with your wish to have your research recorded in our bulletin. Permit me to convey to you my sincerest respects. Do. Louis Lapicque, Honorary Professor, University of Paris. Member, Academy of Sciences. I withdrew my consent for publication in the Bulletin of the French Academy of Sciences on the following grounds. The physical-chemical interpretation would have obscured the biological character of the experiment. During 1937, I produced bion cultures that were confirmed experimentally by Professor Dutil and Nice. This decisively important fact would not be published. The resume proposed for publication in no way represented the detailed report I had submitted to the Academy. Its publication could only have led to misunderstandings, and unsuccessful control experiments would have been the result. Soft iron filings are the most suitable metallic substance for our experiment. Only a few minutes after introducing sterile filings into our standard bouillon KCl solution, delicate vesicles develop from the iron particles. This process can be followed microscopically. A single iron particle is placed on a slide and a small amount of potassium chloride added. Within a short time, bions are produced whose motility lasts only approximately 10 minutes. Like tiny magnets, they order themselves along lines of magnetic force and cling to each other. Cf. Figs. 27 and 28, Appendix. A solution of iron bions becomes colloid within a few days. The particles consist of heavy, angular, intensely blue energy vesicles which become progressively softer and more elastic. Cf. Figure 3. The blue vesicles can form cultures, but this subject will be discussed later. Humus is composed of mostly motile vesicles having an intense blue glimmer. Soil subjected to autoclavation disintegrates completely into energy vesicles. The progressive disintegration can be observed microscopically each day. Cf. Figure 29, Appendix 2. The question of Brownian movement. A few fundamental problems must be cleared up before we can draw any conclusions from our observations. The concept of Brownian movement has been invoked as an objection to the claim that specific bioenergetic forces account for the motility of bions. Physicists have known for a long time that the smallest colloidal particles are in motion, that is, that they move in the field in various directions. These movements have even been calculated. They are attributed to collisions between the molecules in the solution and the larger colloidal particles. This interpretation is purely physical and mechanistic. Nothing in it is consistent with the biological energy manifestations of pulsation. Can this interpretation be applied to the phenomena observed in bions energy vesicles? An interpretation is valid only if it makes new phenomena comprehensible. It is invalid if it conflicts with the observations, and when it directly contradicts the observations and can be replaced by another interpretation that offers a more satisfying explanation of the phenomena, it is useless. The mechanical Brownian movement is defended by physicists as a dogma. Insofar as it is directed against mystical interpretations of living phenomena, this defense is justified. But experience shows just as clearly that the molecular movement interpretation is itself not without irrational motives. Otherwise, the physicist who sees all around him only Brownian movements of a purely physical nature would not so stubbornly refuse to consider a few facts that contradict his interpretation in certain instances. I do not believe I will ever be able to convince these physicists, but I know that the blind alleys into which the purely mechanistic viewpoint leads will one day force science to face up to new facts and arguments. Doubtless, there exist movements of extremely fine particles that allow a mechanical interpretation. For example, I myself believe that the movement of the vesicles, cf. Figure 2, back and forth in place is not of a biological nature. Whether molecules are moving them back and forth I do not know, since I have never seen molecules, any more than have the proponents of the purely mechanical Brownian movement. Now, let us clarify what the physical-mechanical interpretation advocates. Since neither the particles nor the molecules ever disappear in the solution, the molecular impulses should, logically, continue indefinitely, as should the movement of the particles. In addition, all particles in approximately the same size range would have to be in motion. Finally, the only type of movement possible under these circumstances would be from place to place. Contraction and expansion of the contents of the particles cannot be explained by the mechanical interpretation. How could an impulse from a molecule outside the particle cause vibration or an expansion inside? Later, we will become familiar with other properties of the bions that could not conceivably be explained in mechanical terms. These observations can be seen only at magnifications of at least 2000x. This is the absolute minimum requirement. Actually, reliable conclusions require a magnification of 3000–4000x. Just as indispensable is the microscopic examination of a living preparation before it is destroyed by biological stain. The Copenhagen biologist A. Fischer became very upset and even somewhat hostile when he was unable to achieve a magnification greater than 1500x. As I had insisted he must in order to be able to see what I claimed as fact. The stain of the bions done at his institute in 1936 was positive. Under the correct conditions, the following phenomena, which cannot be interpreted mechanistically, manifest themselves. Movement is sometimes present, sometimes absent. It occurs, then stops. Bion vesicles appear at the margin of coal or moss particles and exhibit movement once there is a sufficient degree of tension and charge. Certain Bions preparations show no movement at all. What has happened to the molecular impulses in such cases? The molecules have not gone away and neither have the particles. And mechanistic science does not postulate a third factor in the origin of the movements. The external motion of the energy vesicles must therefore be related to their inner charge. The internal motility of many bions, their expansion, contraction, vibration, and glimmering, is a fact that cannot be ascribed to external impulses, only to shifts in internal energy. Thus the inner motility must also be connected with internal charge. Bion research comprehends living red blood corpuscles as organotic vesicles. Examined at a magnification above 2000x they are blue and pulsate. Dead red corpuscles are not blue but black. They are non-modal and do not pulsate. The motility of red corpuscles originates in the internal charge only, not in external impulses. With the disappearance of the blue, organotic color, motility also ceases. The fundamental question of all biology concerns the origin of the inner impulses in the living organism. No one doubts that the living is distinguished from the non-living by the internal origin of the motor impulses. The internal motor impulse can be ascribed only to an energy active within the organism. The question of the origin of this energy itself is answered by the Bion experiment. The biologically effective energy, within the organism, that generates the impulses originates from the same matter as the bion. I introduced the term orgone for the energy observable and modal. Bion is matter, deriving it from the words organism and orgastic. Henceforth, the expression organotic encompasses all energy phenomena and processes specifically pertaining to the energy governing living matter. Every living organism is a membranous structure containing a quantity of orgone energy in its body fluids. It constitutes an organotic system. Moreover, the purely physical mechanical interpretation fails to clarify a single one of the specific biological reactions. We have freed ourselves from any suspicion that we are dealing with a supernatural life force transcending energy and matter. Therewith, we acknowledge a connection between the energy vesicles and Einstein's functional theory of matter and energy. We have observed some fundamental processes that indicate the manner in which orgone originates from matter, specifically, the processes of disintegration of matter and swelling of the disintegrating particles. The solution to the enigma of the way life functions is contained in these processes. The essential objective functions of biological energy correspond to the essential functions of living matter. The basic functions of highly developed organisms are the same as those of the smallest bits of contractile plasma. Every mechanistic or chemical approach fails completely here. It is not a matter of substances, but of biological energy functions. In this viewpoint, we are in accord with many biologists. For instance, Reich's animal biology today owes its existence to the introduction of physiological experimentation into the study of lower animals. In these experiments the physiologists' expectations of new research horizons were not fulfilled. The breaking down of living phenomena into chemical and physical processes did not advance matters at all. For all those scientists who see the essential element of biology in the life process itself and not in its reduction to chemistry, physics, and mathematics. Umwelt und Inhalt der Tiere, Berlin, Springer, 1921, p. 23. The T. bacilli. In my account of the experiment on coal Bions, I mentioned the discovery, using Gram-stain, of tiny bodies, shaped like lancets. These bodies were given the name T. bacilli, that is, toad's bacilli, because of their dual connection with the process of dying. T. bacilli develop from the degeneration and putrid disintegration of living and non-living protein. Infected in strong doses, T. bacilli are capable of killing mice within 24 hours. If Staphylococcus cultures or rot bacteria, B. proteus, etc., are allowed to stand for a sufficient length of time, a greenish margin forms around the edge of the culture. Against the light, this margin is seen to have a blue glimmer which tends to spread. At the beginning of the experiment, we established that the culture was pure, containing nothing but staphylococci. After a few weeks or months, we take a sample from the bluish-green margin, and find that although there are now very few cocci, the culture is swarming with a variety of much smaller bacilli moving in lively zigzag patterns. These are roughly 0.2–0.5 micron in length, and examined at a magnification of at least 2000x, appear slightly oval-shaped. Cf. Figure 4c. Inoculated in bouillon, they develop a culture fluid with a strong blue-green glimmer and an acrid, ammoniacal odor. The longer the bouillon culture is allowed to stand, the denser it becomes and the deeper its blue or green-blue color. After a few days, rot bacteria cultures, B. proteus, B. subtilis, and staphylococci, cf. Figure 4b, agglutinate at the bottom of the test tube or as a membrane on the surface. The agglutination of the T. bacilli, on the other hand, does not occur for months. In the case of a mixed culture, all other bacilli agglutinate very rapidly, whereas the T. bacilli remain alive. T. bacilli can be obtained from degeneration of every kind of protein substance. To date, T. bacilli, identical in form and 1.4 actions, have been cultivated from 15 different sources. T. bacilli are therefore the product of degenerative putrid processes and ensues. Following is a list of some sources of blood of cancer patients. T. bacilli can be cultivated from the blood of patients with advanced cancer by simple inoculation in bouillon. In our laboratory, this process became one of our most important tests for cancer. Cancer tissue, every kind of cancer tissue, fresh or old, shows T. bacilli on microscopic examination, and yields T. bacilli cultures in bouillon and on agar. When boiled, it disintegrates almost totally into T-bodies with the characteristic red-gram stain reaction. Precancerous cells in tissue, these also yield T bacilli, that is, they disintegrate into T-bodies or already contain them fully developed. Epithelium from the vagina, tongue, skin, or from sputum is normally without structure. In the precancerous state, it shows extremely fine T-bodies on dark field examination. Degenerating blood, experiment, 2 to 3 cc of blood are spread on a sterilized petri dish and dried for 24 hours in an incubator. The dried blood is then dusted with blood charcoal that has been heated to incandescence. After a further 24 hours, enough potassium chloride and bouillon are added to cover the substance. Microscopic examination and biological stain reaction immediately confirm the presence of T bacilli. T bacilli of every origin generate cancerous, destructive, and infiltrating growths in healthy mice. However, I would like to deal separately with this element of the Bion experiment and restrict myself here to the essential, biologically significant reactions pertaining to the problem of orgone energy. Bion preparations regularly yield two types of bions, the blue bions described earlier and the small black T bacilli. These two types are antagonistic to each other in the biological experiment. The bions are capable of killing or immobilizing the black T bacilli. This process occurs in the drop under the microscope as well as in the living mouse, cf. Figure 4a. We place a drop of solution of earth and iron or coal bions on a hanging drop slide and add a small drop from a T bacillus culture. At 400x in the dark field, or more clearly at 2000x in ordinary light, we can see that the T bacilli in the vicinity of the blue bions become agitated, spinning around and around, then remain on one spot, quivering, and finally become immobile. In time, more and more T bacilli accumulate around the blue bions, they agglutinate. The dead bacilli seem to attract and to be lethal to those still living. The orgone energy experiments with cancer had their origin in this significant fact. Subtle as or Proteus bacilli, which are 5 to 8 times the size of T bacilli, are affected in the same way. In these organisms, the lethalness of the blue bions can be observed much more clearly. Ultimately, the whole field is covered with dead bacilli. Between January 1937 and January 1939, injection experiments with bions and T bacilli were carried out on 178 healthy mice. The following table shows the results. Of the 30 T mice that died within 15 months of the T bacilli injection, 25 were carefully examined for cancerous growths. 7 mice revealed emboid cancer cells in various tissues, 13 revealed destructive, infiltrative, cellular growths of a cancerous nature. The remaining 5 had chronic inflammatory growths. The cancer tumors were furthest advanced in the mice that had survived the T bacilli injection for the longest period. The connection between T bacilli and cancer is of crucial importance and will be discussed later. For an evaluation of orgone energy, the results of the experiments have the following significance. 1. Theoretical, at the basis of the life process. At the boundary between non-living and living, we encounter a way of functioning that is completely encompassed by our generally valid scheme of biological functioning. All life functions obey the natural law of the dissociation of the unitary and the functional antithesis and unity of the dissociated out of non-living matter A, modal bions B, containing orgone energy, develop. These bions divide into two groups, the Pa group and the T group. The two groups have an antithetical relationship in that the Pa bions paralyze the T bions. There is only one plausible explanation of this fact in the context of our knowledge of the physical functions of orgone energy. The Pa bions are fully developed, highly charged orgone units. T bions, on the other hand, represent products of degeneration, appearing when tissues, cells, or bacteria begin to lose their orgone charge. They contain only small quantities of orgone and therefore are very weak orgonotic systems. Since the stronger orgonotic system always attracts the weaker and draws off its charge, the paralyzing of T bions by Pa bions is easily explained. The biophysical functional connection between Pa and T is derived from the purely physical functions of orgone energy. 2. Practical, the T are produced either by degenerative processes, that is, through loss of orgone energy in highly developed forms, or they originate because of an insufficient amount of mass-free orgone inside the energy vesicle. T bacilli were produced in my laboratory from the degeneration of the following tissues and bion preparations: dried blood, dried feces, autoclaved cancer tissue, autoclaved egg medium that had been dusted with incandescent charcoal covered with potassium hydroxide, tongue epithelium damaged by dental bridge work, degenerated vaginal epithelial cells, degenerated spermatozoa, degenerated rot bacteria, cold bion preparations of all kinds, and from egg medium treated with potassium cyanide. The deficient orgone content of T bacilli manifests itself in a strange phenomenon which I would like to designate as orgone hunger. The observations are as follows. We filter blood that has been diluted about 10 times with the usual potassium chloride solution. A Birkenfeld filter with a pore size of not more than 0.25 micron catches whatever T bacilli might be present. Microscopic examination at a magnification of 200,400x reveals only fluid, with no structures at all. We add to the serum a tiny drop of pure T bacillus culture, containing no Pa bions. Within a few minutes, an extremely exciting spectacle unfolds, one which in all probability holds the secret of immune bodies in the serum. This phenomenon, known as the reverse ergonomic potential, is discussed elsewhere from a purely physical standpoint. Orgone energy vesicles, bions. At first, we see only T bacilli moving around in the field. Before long, however, large blue vesicles appear here and there, with more and more T bacilli surrounding them, exactly as in a mixture of Pa and T. The T seem to have stimulated the formation of the blue Pa bions. A control serum without T does not show any Pa. The Pa bions, once formed, exercise a paralyzing effect on the T, which begin to agglutinate. A second, even more astonishing process now begins. The heap of agglutinated T is black. There is no evidence of blue. But within 15 to 30 minutes, this dead black heap begins to take on a strong blue coloring and to form membranous vesicles. They are nothing other than Pa bions. The dead T have drawn off orgone from the serum and transformed themselves into Pa bions. These two phenomena have not yet been thoroughly studied. On the one hand, they are connected to the immunity factor and, on the other hand, to the vesicular structuring or the vesicular disintegration of tissues that come into contact with the T bacilli. A few days after a healthy mouse is inoculated subcutaneously with T bacilli, a non-purulent swelling appears on the skin, which, under microscopic examination, proves to be tissue in a state of vesicular disintegration. By the same process, the degeneration of epithelial cells in cancer is marked by the appearance first of bacilli, then of large numbers of blue bions in the surroundings. At this point I would like to interrupt the account of these observations, which yield so much information about organismic orgone energy, and await the results of further experiments before deciding what conclusions to draw. Nevertheless, it is certain that 7% bacilli stimulate Bions disintegration and that cancer cells are organized from bionously disintegrated tissue, just as amoeba and other protozoa are formed from moss bions. The T-blood test the biological vigor, that is, functioning power, of a cell is determined neither by its structure nor by its chemical composition. Disintegration of structure and chemical composition must be regarded as consequences rather than causes of the biological degeneration. The structure, along with the biochemical equilibrium of the cell, is the expression of the cell's biological vigor, but the biological function itself has hitherto been a mystery. The organotic charge of the cell now provides us with the possibility of determining it experimentally. The red blood corpuscles of two persons can be alike in structure and chemical makeup while differing sharply in biological function. Under the microscope, both may have the same form. The red corpuscle count and the hemoglobin content may well be normal and identical for both persons. Let us now expose blood samples from each person to the same destructive agent. We autoclave a few drops of blood from each person in bouillon and potassium chloride for 30 minutes at a temperature of 120 degrees Celsius. With steam pressure 15 pounds per square inches, microscopic examination may now yield two very different results. The autoclaved blood of one person has disintegrated into large blue bion vesicles. The blood of the other person yields no blue vesicles, only T bacilli. Gram stain confirms this difference. One blood sample produces blue, gram-positive vesicles, cf. Figure 31, Appendix, the other, red, gram-negative T bacilli. For T bacilli from a sarcoma, cf. Figure 32, Appendix. This conclusion may be drawn: the former blood sample shows a strong organotic charge of the red cells. The charge reveals itself, after autoclavation, in the blue bions, B reaction. The latter blood sample shows a weak or minimal orgone charge of the red cells. The lack of organotic charge manifests itself, after autoclavation, in the absence of blue bions and in the presence of T bacilli, which result from the degeneration of the red corpuscles, T reaction. The T reaction is typical for cases of advanced cancer in which the orgone content of the blood has been totally consumed and the organisms struggle against the systemic disease, cancer biopathy, and the local tumor. This T reaction is usually present before any symptoms of anemia and often reveals the cancer process long before a perceptible tumor has formed. On the other hand, red corpuscles weaken orgone energy, absorb it greedily when it is supplied to the organism by the organic emulator. Subsequent autoclavation tests yield a shift from the T reaction to the B reaction, that is, the red blood corpuscles have become more resistant to autoclavation, they contain more orgone. The red corpuscles can be charged by atmospheric orgone energy. The effects of solar radiation are based on the same principle. This can be confirmed experimentally. On the microscope slide, we mix biologically, that is, Organotically, weak blood with rot bacteria or T. bacilli, the blood, being energetically weak, does not destroy or agglutinate the bacteria or the T. bacilli. However, once the organism has been organotically charged, the degree of charge can be ascertained by means of the autoclavation test. The blood has a decidedly paralyzing and agglutinating effect on the same pathogenic microorganisms. Smaller protozoa respond in the same fashion, that is, they are not damaged by organotically weak blood but are paralyzed by organotically strong wood. The red corpuscle is an organotic system in miniature, containing a small quantity of Orgone energy inside its membrane. At 4000x magnification, the red blood corpuscles show a deep blue glimmer and lively vibration of their contents. They expand and contract and are therefore not rigid, as is usually thought. They carry atmospheric Orgone energy from the lungs to the tissues. The nature of the relationship between atmospheric oxygen and Orgone energy can only be surmised at this time. Whether Orgone is identical with the chemical particles of the air or fundamentally different from them is unknown. The organotic charge is also revealed in the shape and structure of the red blood corpuscles. Cells with a weak charge are more or less shrunken and have a narrow blue margin which glimmers feebly. Once the organism is charged, the red blood cells swell, while the blue margin intensifies and widens, sometimes including the entire cell. No pathogenic microorganism can survive in the vicinity of these organotically highly charged red blood cells. How these facts are connected to immunity against infectious diseases, colds, etc., is still obscure, but finding the connection should not be too difficult. Probably, the organotic charge of the tissues and blood cells determines the degree of susceptibility to infections, the disposition to disease. The fact that during the process of destroying the pathogenic microorganisms the erythrocytes gradually lose their blue coloration, turn black, and sometimes degenerate into T-bodies demonstrates that the Orgone charge of the red blood corpuscles actually kills protozoa and bacteria. Examination of tissue from tumors and treated mice shows that when charged red blood corpuscles permeate cancer tissue it disintegrates into non-motile T-bodies. In this process, the red blood corpuscles disappear and only T-bodies can be seen. The cancer tumor shows large cavities which are filled with T-bodies, visible on dark field examination at 300–400x. Macroscopically, the content of the cavities at first appears blood red, but gradually turns a rust brown due to hemosiderosis. The iron pigment has separated from the disintegrated corpuscles, which have lost their charge of biological energy. The typical secondary anemia of cancer patients is the expression of the loss of biological energy from the blood in the struggle against the T bacilli and the cancer cells. More details on this subject will be presented later in connection with our cancer experiments. What is important here is to learn how the organotic charge of the blood cells acts under a variety of conditions. To put it briefly, erythrocytes with a strong Orgone charge act upon bacteria and small protozoa in exactly the same way as do earth, iron, coal, and other bions. Since they originate in bone marrow, it must be assumed that the bone marrow has the capacity to generate bions constantly. Energy vesicles are basic to both animal and plant tissue. Taken together, these facts form the basis for the Orgone therapy experiments on cancer patients. The introduction of orgone energy from the outside relieves the organism of the burden of consuming its own body orgone in the struggle against the disease. This is a further proof of the identity of atmospheric and organismic orgone energy. Experiment reveals the following. 1. A grass infusion develops no protozoa, or very few, when kept from the beginning in the organic accumulator. Clearly, orgone energy charges the grass tissue and prevents its disintegration into protozoa. 2. Fully developed protozoa are not destroyed in the organic accumulator. 3. T bacilli are not destroyed in the organic accumulator but, on the other hand, a cancer patient's blood can be cleared of T bodies within a few days if the patient is exposed to intense orgone irradiation. Lumination and attraction. It is a generally known fact, first discovered by Gerwich, that animal blood radiates. Since, from the point of view of orgone biophysics, erythrocytes are nothing but orgone energy vesicles, it is important to demonstrate the radiation microscopically. One of its most salient characteristics, as we discover in pure orgone physics, is orgonotic attraction, which has no connection with iron magnetism. Observation of this orgonotic attraction is made possible by the following experiments, all of which consist of bringing together bions of various origins in the usual preparation of bouillon and potassium chloride and examining them under the microscope. Seen energetically, the destruction and agglutination of bacteria by various kinds of bions is quite simply the attraction and subsequent withdrawal of orgone energy by the stronger orgonotic system, the bion. By mixing different kinds of bions, we discover other important effects of orgone energy. Let us experiment first with a sterile mixture of red blood corpuscles and earth bions. We use a drop of diluted blood and one of earth bion solution that is sufficiently thinned out to permit easy observation of each individual bion. No results can be expected at less than 2000 x. A good 80x, a pochromatic objective lens combined with a 16x eyepiece and a microscope with inclined binocular tube would serve the purpose. The use of a special water immersion lens, which can simply be dipped into the solution, is advantageous. The work is easier and faster with direct water immersion and mechanical interferences are quickly offset. An effect from the metal need not be feared, since the phenomena is the same when a cover glass is used. But at this magnification and with the necessary use of hanging drop slides, cover glasses are inconvenient, since they break so easily. Any control experiment will prove that the immersion of the objective lens into the solution does not affect the results in any way. The radiating bridge between two orgonotic systems. At first, the earth bions and red blood corpuscles move about separately. Gradually, however, grouping begins, with several erythrocytes gathering around one of the larger, heavier, earth bions and moving nearer and nearer until they are touching. At each point of contact, a strong radiation appears. At points where the bodies do not touch each other directly but are separated by approximately 0.5 to 1 micron, a strongly radiating bridge is formed between the earth bion and the erythrocyte, and apparently connects them. This bridge vibrates intensely, becoming alternately wider and narrower. Finally, the membranes between the bodies appear to be less distinct. With sufficient observation, it is possible to see clearly that the erythrocytes refract the light more strongly, that their blue color becomes more and more intense, and that they become larger and tauter and show a lively pulsation. In this manner, Erythrocytes can be organotically charged just as effectively as the body is charged by organoradiation of the organism. If weak, deformed erythrocytes from the blood of cancer patients are used for this experiment, their expansion and radiation are more evident. Organotically weak erythrocytes exercise little or no influence on bacilli and small protozoa, but they do become effective when they are charged with orgone. The erythrocytes drink their fill of orgone from the earth bions. The injection of sterile earth bions into cancer mice achieved the same effect as exposure to orgone radiation in the organic emulator: inhibition of the growth of the tumor, replacement of the tumor tissue by strongly radiating blood, and destruction of the T bacilli. What actually occurs in the organism as a result of the bion injections can be seen in the microscopic study of the bion mixture. Bion injection was the method of orgone application utilized in my laboratory before the discovery of atmospheric orgone energy, after which it was replaced by irradiation in the organic emulator. There is no fusion of earth bions and erythrocytes, only the formation of the radiating bridge. This is also true of iron bions, coal bions, etc. On the other hand, coal bions and bions produced from autoclaved blood or from any protein do interpenetrate. This fusion is extremely significant for an understanding of the experimental production of tumors in mice by means of tar. 5. Fusion and interpenetration. Sexuality has in common with procreation the fact that two living systems fuse organotically. In single-celled organisms, mutual penetration of body substance follows the fusion that occurs in copulation. Fusion and interpenetration are not only energy processes but processes that involve substance as well. In the metazoan, however, these processes are much more complete in terms of energy than of substance. In the copulative act, the male organ penetrates the female. Both organs now form a functioning unit. In many hermaphroditic mollusks, snails, worms, penetration is mutual but restricted to the genitals. The union of two gametes to form a zygote, on the other hand, is a perfect example of total interpenetration and fusion of substance. Although in multicellular animals fusion is restricted to the genital organs and the reproductive cells, the organotic function is total, that is, in copulation, two beings fuse temporarily into a single organotic energy system. In the case of humans, this is true only if both partners are orgastically potent. Organotic fusion is regularly preceded by humanation. Orgonadiocell lumination, physiological excitation, and psychic sexual emotion are functionally identical processes. Psychic perception of a sexual object can cause physiological excitation, erection. Conversely, physiological excitation, stroking, friction, can evoke sexual emotion. This psychosomatic process leads to organotic lumination of the entire organism. On the other hand, strong organotic lumination in an organism tends to intensify the urge for physiological friction with accompanying psychosexual emotion. Therefore, our diagram of biological functioning is again valid. The concept that energy manifestations of the multicellular organism are present in each individual cell and, strictly speaking, have their origin there must be taken seriously. Orgone lumination of the individual cell is identical with the physiologist's and biologist's cell excitation and the depth psychologist's organ libido or cell libido. We have taken an important step toward the understanding of these processes, since we now know that a demonstrable, Specifically, biological energy is active in the cells. The principal energy functions are formation of a radiating bridge, sexual contact, lumination, fusion, and interpenetration. Our purpose here is simply to observe the energy processes of fusion and interpenetration as they are revealed in the Bions. Our sense of triumph when we witness the fusion of a cold bion and an earth bion is justified. The hypothetical concepts cell excitation and organ libido have become tangible realities. After the cold bions are attracted to the heavier, and therefore less modal, earth bions and the radiating bridge is formed, the energy process continues to parallel every phase of copulation. The excited, that is, luminator egg, bions move ever closer to each other. At the point where the radiating bridges appear, the coal substance, in bion form, gradually begins to penetrate the earth bions. One has the impression that the earth bion absorbs the coal bion. Eventually, the smaller coal bions completely penetrate the earth bions. Their black membranes are clearly distinguishable from the brownish membranes of the earth bions. As a whole, this mixture of earth bions and coal bions appears brownish-black. Then, gradually, the black disappears, the membranes of the coal bions dissolve. The earth bions take on a darker coloration and their blue vesicles emit a stronger radiation. Finally, all traces of the coal substance vanish. Another way of describing this process would be to say that the earth bion eats up the coal bion. But it would make no sense to subdivide fusion into eating or copulating since, at this most primitive biological stage, biological energy functions without differentiation. It would be difficult to differentiate functionally between copulation and the incorporation of smaller protozoa by larger OBs. This statement is equally true of the multicellular animal when we describe energy processes and avoid the anthropomorphic trap of ascribing purpose. A suckling child is fulfilling a completely different purpose than is a woman who takes the male organ into her vagina. The first act serves the preservation of the individual, the second the preservation of the species. But these finalistic distinctions are incorrect when the problem is one of biological functioning. In terms of energy, and, biophysically, no other view is valid. The process taking place between the mother's breast and the baby's mouth is precisely the same as that between the erect penis and the vagina. This functional identity extends to the most minute physiological detail. Moralistic hypocrisy should not be permitted to obscure the significance of these matters by representing the holy suckling process as asexual so as not to profane it by identifying it with the devilish, dirty, sexual act. It is not a matter of holy or unholy, but of biological functioning. We only now understand biophysically Freud's fundamental discovery that the nursing child's mouth is just as much a sexually excited organ as is the mother's nipple. In excitation. This one insight will help more in diagnosing the inability of some women to breastfeed their children than all medical theories. It is therefore a matter of indifference to us whether the earth bion has eaten up the coal bion in order to strengthen itself, thereby behaving in a moral or rational manner, or whether it has fused sexually with the coal bion. Instead, we are concerned with the energy functions, which are fundamental in the process itself and common to all basic biological functions, whether we are considering ingestion, conjugation, copulation, the formation of zygotes, or the sexual act of the metazoon. It should be noted that the orgone hunger of bions does have a satiation point. If a limited number of coal bions are introduced into a solution of earth bions, after a few days no coal bions are left. But if a large number of coal bions are added, not all of them disappear. Different species of bions display varying degrees of orgone hunger. The behavior of sand bion cultures toward coal bions is, for instance, gluttonous. Iron bions also fuse easily with coal bions. Bions formed from cooked organic substances, such as mussel, are far less greedy in their absorption of coal bions. This indicates that the smaller the original amount of carbon contained by a bion, the stronger its tendency to take in carbon. Sapa bions, originating from sand, initially contain no carbon. Iron bions contain only traces of it. Mussel bions are, however, composed of carbon compounds. Their hunger for carbon is therefore far less than that of sand bions. I shall refrain from drawing far-reaching conclusions about the origin of plasma on our planet and hold strictly to the facts. Coal bions are not the only ones that fuse with others. Fusion also occurs between earth bions and iron bions and mussel bions, etc. Here is a broad field for fruitful research. To summarize, Orgone energy vesicles show the basic functions of completely developed living substance: attraction, illumination, a radiating bridge, fusion, and penetration. These functions are specific properties of the Orgone vesicles. They are absent in bions that have lost their orgone charge. These functions are therefore determined not by substance but by energy. They are specific functions of the orgone and have nothing to do with magnetism or electricity. We are now better prepared to approach the observations provided by our investigation of the organization of protozoa and cancer cells. 6. The natural organization of protozoa. I would like to begin this section with the excellent statement by the biologist Huxley, taken from the foreword to his book Umwelt und Innenwelt der Tiere. Today, the word science has become a ridiculous fetish. It therefore seems appropriate to point out that science is nothing more than the aggregate opinion of living scientists. In time all opinions are forgotten, rejected, or changed. For that reason, the question what constitutes a scientific truth can be answered without exaggeration by saying an error of today. We hope to progress from gross to smaller errors, but whether we are really on the right track at all is a matter of extreme doubt so far as biology is concerned. In the summer of 1938, I published in Die Bion a number of photographs taken with fast motion techniques and time exposures. They unequivocally demonstrated that single-celled organisms do not evolve from mere bacteria that no one has ever seen but rather from bionously disintegrating moss and grass. The world of biology kept silent on this error of today and with just a few exceptions made no comment on the microphotos. Today, the facts speak for themselves. Cf. Figs. 34, 35, 36, 37, 38, 39, 40, 41a, b, c, 42, Appendix. The animal is not a mechanical sum of organs, just as the organ is not a mechanical sum of cells. The animal in its totality, every organ and every cell, individually and collectively, is a self-contained organotic system characterized by the 4-beat pattern of tension-charge-discharge, greater than relaxation, and thereby a functional unit. It is a property of the jiving system that each one of its parts can operate according to the tension-charge formula independently or in connection with the organism in its totality. In modern biological literature, since approximately World War I, the functional approach has slowly been making some headway against the substantive mechanistic viewpoint. One of the crucial problems of biology is the fact that whereas the functions of structures, musculature, nerves, glands, etc., are understandable mechanically, the functions of protoplasm are treated as though they were a miracle. Protoplasm is fundamentally different from a machine in the very fact that fluid machines are inconceivable. T.Lksky 11. Protoplasm functions on the basis of characteristics not possessed by machines. It functions without being structured. It is preserved by a balance between assimilation and assimilation, which is to say through function and not through material structure. When function ceases, the material state falls apart. The substantive structure is itself dependent upon the function of the living plasm. The animal is an event, Jennings. These facts are inaccessible to the mechanistic materialistic orientation of biology. What answer, then, does knowledge of orgone functions give to this riddle? One thing is clear, any concept that deduces everything from germline, while assuming the eternal existence of germplasm without investigating its origin, cannot enlighten us about plasmatic functioning. Such a concept, which sees everything to be already materially existent in the germ, is similar to the old notion of the preformation of all future generations in the germ cells. If, now, the transformation of matter originally non-plasmatic into plasm can be observed microscopically and on film, it is possible to arrive at conclusions about the development of plasmatic function itself. We can, without hesitation, consider plasmatic function as identical with the function of tension and charge. For all complex functions can be traced back to the alternation of expansion and contraction in the biological pulsation. Non-living substance does not pulsate. Living substance pulsates. The solution of the enigma of biogenesis should be sought precisely at the point of transition between non-motility and pulsatory motility. This transition point can, in fact, be observed microscopically and recorded on film. The damage that metaphysical germ theory has done in biological research can be assessed by the following facts. Not one single biology textbook known to me contains a description of the material of protozoal infusions. We learn nothing about what is actually happening in the grass or moss. The protozoa are represented as simply present, a finished product. The argument that no protozoa exist in moss that has been heated, so often raised against the concept of the natural organization of single-celled organisms, cannot be taken seriously. Its inadequacies were pointed out by biologists a long time ago, and the stubborn adherence to it only betrays an interest that is not scientific but religious. We can now state that if heated moss contains no protozoa, it still contains blue Bions. 22 in 1944, protozoa were obtained from grass that had been sterilized at 50, 60, 70, and even 80 degrees centigrade. However, such Bions are also to be found in unheated moss, which undergoes a process of gradual swelling. Every authoritative book on biology contains the assertion, blindly copied by one author from another, that protozoa evolve from encysted germs. Protozoa can become spherical in shape, it is observed. The observation is correct, of course, but the interpretation of it is erroneous. Two separate processes are being treated as though they were one. One process is the assumption of a spherical shape by protozoa when they are damaged. The other process involves the evolution of spherical shapes in grass infusions that have no connection with the spherical protozoa. These spherical forms are the result of a gradual development of bions into a heap of Bions vesicles. The spherical heap of bions represents a typical stage in the natural formation of single-celled organisms. Biology textbooks claim that the infusions are full of algae, which in turn are supposed to have evolved from germs. It is true that protozoa take their nourishment from these algae. Yet, who has ever seen the germ of such algae? And how does it happen that these so-called algae can be found in large numbers, motile, in preparations consisting of only autoclaved blood or muscle tissue? The algae are nothing but our Bion vesicles, into which every kind of organic tissue disintegrates on swelling. There are no protozoal germs in the air, Pouchet. Biologists' notions to the contrary will appear to us no less remarkable than the phenomena that we ourselves are about to see. For while others are busy maintaining that protozoal germs are to be found in the air and that they settle down to start their evolutionary process wherever they find a suitable culture medium, we will take the trouble to observe an infusion microscopically for an honka day from the moment of its preparation, at a magnification of 2000x. As a preliminary, we conduct a simple experiment to see if we cannot perhaps find some of these spores from the cosmos which have drifted down and settled upon blades of grass or moss. Holding a few blades of grass and a pincette, we scrape germs into a glass of unsterile water. The grass has been examined to see that the grass has been examined, is not already in a state of vesicular disintegration but still has its usual cellular structure. No matter how we conduct the experiment, not a single protozoan nor a single cyst is to be found in the unsterile water into which we have dipped the grass blades. What processes, then, are at work in the infusion? Over the course of 2 to 3 days, the grass blades undergo vesicular disintegration, as does every other substance subjected to the swelling process, cf. Figs. 34, 35, Appendix. Even at a magnification of 4000x, there is not a trace of bacteria, cysts, or protozoa. However, the grass blades disintegrate more and more. After an additional 2 to 3 days, there is scarcely a blade left that still retains its original cellular, striated structure intact. Individual protozoa may well be present, but we direct our attention to the bions. Here and there, the Bion vesicles can be observed to gather into heaps surrounded with membranes. Each step in this development can be followed. Occasionally, within a heap, individual Bion vesicles begin to show delicate rotating or vibrating movements. They become increasingly taut and begin to look like cysts. However, they are not dried-up protozoa but forms in the process of developing from Bion heaps. These Bion masses, or heaps, take on a variety of sizes and shapes. The tauter they are, the more spherical they become. They have filled with fluid, and are thus mechanically tense. The first stage of the tension charge function is complete. It is worth the trouble to examine a single formation for many hours at a magnification of 2000x, replenishing the fluid to keep it in focus. We can film this one formation in fast motion, which makes the work easier, but many interesting details are lost. The development of such a Bion heap into a pulsating, single-celled organism takes 1 to 2 days. The protozoal germ vesicle, the Bion heap, remains immobile for hours, but as the fast-motion film shows, it becomes tauter and more sharply delineated from its surroundings. Gradually, a movement of the energy vesicles begins to set in within the bion heap. The following types of motility can be distinguished. 1. Rolling. The energy vesicles inside the bion heap roll rhythmically toward and away from each other, creating the impression of mutual attraction and repulsion. The causes of this rolling movement can only be surmised. Initially, the vesicles of the disintegrating grass firmly adhere to each other. Their Orgone charge is the same as that of the grass bions that do not develop into protozoa. As the spherical heap of vesicles swells and becomes tauter, more Orgone has to be developed inside the individual vesicles, since, as with the cold bion, the swelling thins out the membrane and intensifies the Orgone charge. Once the vesicles inside the bion heap have lost their mechanical adhesion to each other, the Orgone charge can begin to express itself in motion, with the subsequent development of orgonotic attraction between the vesicles. The attraction effect exercised by the more remote vesicles results in mutual attraction and repulsion. -2. Rotation. In many bion heaps the entire contents of the vesicles begin to rotate in one direction. This movement can last for hours. It increases in intensity until, in the end, the whole mass, including membrane, is rotating. During this process, the membrane separates from the surrounding grass tissue. 3. Confluence of the energy vesicles. Not all bion heaps retain the vesicular structure of their plasm. In many kinds of amoebae, the boundaries between the individual energy vesicles disappear, and the plasm then forms a homogeneous mass with a blue glimmer. In others, the vesicular structure persists throughout the complete development. This is true also for cancer cells, which organize themselves from disintegrating animal tissue by precisely the same process as that by which protozoa are formed from grass or moss. Cancer cells are nothing but protozoa that form in the animal organism from tissue bions. The natural organization of protozoa in grass or moss. Infusions is the key to understanding the organization of cancer cells in animal tissue. Of cancer cell. 4. Pulsation. At a magnification of about 3000x, extremely fine movements of expansion and contraction can already be seen in the bion heap. Those forms in which the energy vesicles coalesce are apparently much more capable of pulsation than those which retain the vesicular character of the plasm, as, for instance, the protozoan, Vorticella, cf. Figs. 39-42, Appendix. A variant of the confluence of bions can be seen in the primal vesicles of many Pyromachia. In this case, the small bions do not converge together into one mass, but instead form vesicles of medium size, and group in the interior of the larger body. The vesicles then start rolling and rotating movements around and toward each other, just like the bions from which they originate. The more bions flow together, that is, the snore fluid the plasm, the more motile is the total organism. In addition to finished animals, evolving organisms and shapes at all stages of development may be seen, principally at the margins of the disintegrating moss or grass. As in the amoebal Ornax and B. Prochus, the nuclei are formed by a distinct concentration of energy vesicles, which is revealed in a stronger illumination. It is difficult for the observer to follow the transition from one stage of development to another because of fatigue. The device for making fast-motion films is an invaluable aid in this respect. For filming over the course of several days, a cover glass should be placed over the hanging drop slide in such a manner that a portion of its cavity remains uncovered. In addition, some fluid should be drawn over the edge of the cavity, forming a reservoir on the outside that can be used continually to refill the preparation without having to disturb the focus. Filming with the cover glass completely covering the cavity is impossible, first because it would cause the formation of air bubbles, and second because it would make the microorganisms suffocate. Filming of the Bions structure and plasmatic motility in Protozoa has thus far been successful at a magnification of 2300x. The fast motion technique allows a process that took 2 to 3 days to complete to be viewed in a matter of minutes. In all Protozoa the development is identical, up to the stage when the Bions primal vesicles are formed. From that point on, the Protozoa are differentiated according to a law not yet apparent. After years of observations, I cannot assume that the initial primal vesicles belong to any specifically individual forms. My own surmise is rather that the differentiation of forms sets in only at a certain point of common evolution, cf. Figure 7. Here, further observation will make it necessary to amplify and correct a great deal. One of the greatest mysteries is why certain forms, once they have developed, reproduce thereafter in the same form. In this connection, the functional standpoint will have to enter into many battles with the metaphysical theory of heredity that substitutes ready-made genes for comprehension. The primal cells that incline toward a rotating movement and possess a large vesicular structure usually develop into permeates. The resting primal cells, in which the bion content liquefies, develop into flowing amoebae, amoebolimax, cf. Figs. 36, 37, Appendix. The way in which both types separate from their matrix is likewise characteristic. The permeates roll themselves free, while the amoebae simply flow away from the bion mass, cf. Figure 38, Appendix. The vorticelli, which I called Protozoa because of their orgasm-like contraction and expansion, often adhere to the grass blade until fully developed, cf. Figure 39, Appendix. But in some cases they detach themselves and swarm around freely, sometimes with a tiny piece of Bions grass adhering to them. When completely developed, the Protozoa absorb Bions from the fluid by attraction. The attraction exerted by permeates and colpodia on the energy vesicles is enormous and cannot be attributed to the mechanical movement of the cilia, for the vesicles in the fluid do not move past the body with the current, as would be expected from the action of the cilia. Instead, once within a certain proximity to the paramecium, they are drawn toward it with great force. The impression is unmistakable. After it has contracted, the protozoan expands and opens wide at the mouth end. The Bions in the fluid then stream into it with great force. The mouth closes, the protozoan contracts back to its spherical shape, and the energy vesicles inside start up a rhythmical, grinding movement. Observation at 2000-3000x reveals interesting information about the origin of the internal movement impulses. With absolute regularity, the vesicles, which have an intense blue color, begin a vigorous rotary movement at the point where a pseudopodium is about to be formed. Then the plasm starts to stream outward toward the periphery, that is, expansion begins. Many of the blue vesicles are swept along in this process, losing their blue color and becoming black. This permits the following explanation: the protrusion of the pseudopodium is preceded by a rapid, intense concentration of orgone, which is the source of the expansion impulse. If the impulse is expressed in movement, then, clearly, orgone energy is being used up. There is no other explanation for the fact that the vesicles become black. I would not care to generalize this correlation, since the phenomenon just described is not to be found in all protozoa. There can be no doubt, however, that the energy for movement, in the form of expansion impulses, is provided by the orgone charge of the bions and the protozoan. The mechanistic interpretation based on Brownian movement fails completely here. Equally inadequate is the chemical concept of plasma as merely a highly complex carbon substance. Living protoplasm is a complex protein governed by mass-free orgone. The body of the protozoan shows an orgone energy field which influences its surroundings. It affects bions, small bacteria, and other small protozoa, mostly by attracting or paralyzing them. Charged erythrocytes seem to be organotically stronger than pyromachia and amoebae, since they are capable of limiting the motility of these organisms. From the standpoint of orgone physics, then, the protozoan consists of a nucleus, a plasmatic periphery, and an orgone energy field, which together comprise an organotic system. It should be pointed out that the origination of protozoa from Bions plant tissue does not contradict their procreation through division. Development by natural organization and procreation through division occur simultaneously, as can easily be observed microscopically. There is probably nothing more suitable for the study of the functions of tension, greater than charge, -4 discharge relaxation than protozoa. Their movements, their plasmatic currents, their expansion and contraction speak in entirely unequivocal language in terms of our formula of the life function. By using weak currents of electricity it is possible to change, accelerate, and even destroy these functions. But, as the development of the protozoan shows, the energy involved in these functions emanates from the bions that compose the protozoal primal vesicles. The locomotor functions of the amoeba must be ascribed solely to orgone energy, in view of the lack of all structural composition. The appearance and disappearance of pseudopodia are expressions of the expansion and contraction functions. The amoeba does not form a pseudopodium for the purpose of reaching an object, that is, on the basis of a goal, finalistically speaking. It does it functionally, that is, when a suitable object, by means of attraction, provokes an expansion impulse in the organotic plasma. If only to protect future research, it is essential to maintain the principle that living matter simply functions, without sense or purpose. If one resorts to purposes, everything is easily explained. It is much more difficult to discover the nature of unknown functions. The purposeful behavior of the amoeba can be reduced to the function of orgone energy. For example, it incorporates a bion vesicle because the stronger orgone charge of the protozoan attracts the weaker charged free bion. It may be correct to say that the purpose of nourishment is thereby fulfilled, but this fulfillment is an effect, not a cause, of the act of ingestion. The protoplasm does not function according to mechanical, or metaphysical, finalistic principles, but on the basis of organotic energy functions. In principle, all biological functions can be reduced to pulsation, that is, to the alternation of expansion and contraction. Pulsation itself is a natural expression of the two antithetical functions of Orgone energy, the dissociation and attraction of Orgone-charged organic matter. On the basis of the processes that occur in the plasm of the amoeba, we must assume that the immediate state of matter has the following influence on the function of Orgone energy: the individual vesicles, with their Orgone energy content, can arrange themselves in rows, as in moss, and form a striated tissue structure. This process is a product of the organizing function of attraction. With other protozoa, the energy vesicles converge and form a homogeneous mass. Every concentration of Orgone-charged matter, as a consequence of attraction, automatically triggers the impulse for dissociation, thus affecting a separation of the particles. If the amoeba has already taken on spherical shape, then, at the point where the energy vesicles are most densely concentrated, a lively rolling movement appears. This movement evolves into an expansion, that is, a separation of the vesicles. On the other hand, the separation of the vesicles from each other triggers the impulse for rapprochement through attraction. In this way, contraction takes place, as long as the plasm is sufficiently charged with Orgone energy. Every contraction induces an expansion, every expansion a contraction. This inner process appears externally as biological pulsation, whatever its rhythm. Since expansion is accompanied by swelling, and contraction by detumescence, the mechanical and orgonotic functions meet in the 4-beat pattern: swelling, tension, greater than expansion, charge, contraction, discharge, detumescence, relaxation. But pulsation itself is essentially bound up with the alternation of dissociation with attraction and attraction with dissociation of the particles. This process may be seen with particular clarity in the expansions and contractions of the Vorticella or protozoan. After every contraction, a grinding or frictional movement of the vesicles begins in the body, culminating in an expansion. Connected with this expansion is the opening of the mouth end, which causes an attraction of energy vesicles from the fluid, eating, that is, a charging. This is followed by a contraction to the spherical shape, that is, a rapprochement of the inner vesicles. The process is endless. We will encounter this biophysical functioning repeatedly. Biological pulsation, that is, contraction and expansion, the central phenomenon of living substance, is thus explained by the two antithetical, basic physical functions of biological energy, attraction and dissociation. Summary: when matter is exposed to high temperatures and made to swell, it undergoes vesicular disintegration. 2. Although high temperatures, such as heating to incandescence, about 1500 degrees Celsius, or autoclavation at 120 degrees Celsius, destroy life, they produce energy vesicles which may then develop into living organisms. 3. The energy function in Bions originates from the vesicular disintegration of matter, not from an external source. 4. Energy vesicles are infinitesimal quantities of matter that contain a quantum of energy originating from this matter. 5. Bions are carriers of biological energy. They represent a transition from the non-living to the living. 6. The blue color of Bions is a manifestation of this energy. With the disappearance of this color, the basic biological character of the bion ceases. 7. The bion experiments do not create artificial life. They merely reveal the natural process by which unicellular organisms and cancer cells derive spontaneously from the vesicular disintegration of matter, Experiment XX3 on the organization of plasmatic matter from mass-free Orgone energy. In the following pages I would like to report on an experimental finding that owes its discovery to one of those accidents that a protocol begun January 2, 1945. Notarization of the protocol, March 8, 1945. Conclusion of the protocol, May 26, 1945. Often occur in the course of systematic experimental investigations and which when looked at more closely, usually prove to be completely logical consequences of experimental thought and work processes. The occasion in this particular case was a simple experiment, No. 20, 1944 to 1945. For several years, I had been observing Earth bion preparations which I kept alive by continuously replenishing the water. I noticed after a few months, in other cases, only after several years, the appearance in these preparations of small, rapidly moving forms, shaped like bends or the heads of spermatozoa and resembling living organisms. I was able to clearly follow the evolution of these forms from strongly radiating, slowly pulsating bions. As we already know, such living forms do not originate in the air. First, they are not found in air dust. Second, they cannot be cultivated from air infection. And third, their appearance in the non-sterile Earth bion preparations only after months and years confirms their organization from the preparations themselves. In addition, we found that autoclaved preparations, which were kept sterile, yielded the same living forms. In December 1944, we acquired an apparatus designed for the quantitative measurement of the fluorescence of liquids, and also for colorimetry. The use of this apparatus was to be based on the following considerations. Earlier experiments had demonstrated that Orgone energy possesses the property of lumination. It was to be assumed that fluids with a higher organotic potency, which is to say, those containing more Orgone energy, would laminate more strongly than fluids with weaker organotic potency. Accordingly, the intensity of fluorescence in fluids would be considered as an expression of lamination. As a working hypothesis, the degree of fluorescence was made the measure of organotic potency. These assumptions were subsequently confirmed and led to practical and controllable experimental results. Measurement of the fluorophotometric intensity, that is, the organotic potency, of fluids that had contained Earth bions for months and sometimes years showed immediately a far higher fluorophotometric value than that of ordinary water. We now wanted to determine in what way the organotic potency, which we had measured fluorophotometrically, would change under various conditions. After first measuring their fluorometric content, we left several sealed ampoules of Earth bion water in the laboratory itself, and put others in a small 3-layered accumulator, in the X-ray room, in the open air, and buried in the ground. Our only purpose was to measure the fluorometric values again at the end of a specified time. After 3 weeks, we noticed that the ampoules that had been left outside in the open and had frozen contained dense flakes after thawing. We were about to throw away these particular ampoules as contaminated, when it occurred to me to look at the flakes under a microscope. This proved to be a fortunate decision. To my great astonishment, I discovered that the flakes, which had formed in a crystal clear, that is to say, an absolutely particle-free fluid, were, when examined microscopically, intensely radiating particles of bionous matter. At a higher magnification, 3000x, it was possible to observe contracting and expanding bions, with which we were already familiar. We repeated the experiment, filtering and freezing crystal clear Bion water, until all doubt was eliminated. We had discovered a process by which Orgone energy existing freely in water, that is, not bound up in Bion's matter, can organize itself into plasmatic, living substance exhibiting all the criteria of life. In the following account, I shall restrict myself to reporting the technique of Experiment 20 and providing the verified facts. For the moment, I wish to refrain from discussing the experimental data in a larger theoretical context. Their implications are of extraordinary significance, but they are understandable only when considered within the total context of the Orgone physical function. This will be attempted elsewhere. Here, I would like merely to point to the advance that this experiment represents in the preparation of Bions, that is, of viable Orgone energy vesicles. To summarize, 1. From 1936 to 1945, Bions were prepared exclusively from fully organized matter, humus, grass, iron, sand, carbon, etc. The advance represented by Experiment 20 consists of the fact that now Orgone energy vesicles, possessing all the properties of living matter, also can be obtained from mass-free Orgone energy. We may therefore differentiate between the production of Bions from matter already organized, secondary Bion formation, and the organization of Orgone energy vesicles from unorganized energy, primary Bion formation. The significance of this distinction for biogenesis and biochemical problems will be presented elsewhere. 2. Experiment 20 also represents an advance on past Bion experiments insofar as it provides a new, irrefutable proof of the life-specific nature of Orgone energy. The fluorophotometric demonstration of Orgone energy in earth Bion water. 1. Ordinary garden soil is sieved free of stones and jumps of clay. Examined microscopically after water has been added, the sieved earth shows no motility. 2. Distilled water and spring water are examined fluorophotometrically. If the fluorophotometric value of distilled water is set at 1, then water containing salts, that is, spring or tap water, has a value of 3 to 4, as mirrored in Forest Hills, New York. We take the linearophotometry value of a fluid as the expression of its orgonotic lamination, that is, its orgonotic potency. The galvanometer connected to the fluorophotometer has a scale of equidistant graduations from 1 to 100. The orgonotic potency of the fluid measured can then be seen as a multiple of the orgonotic potency of distilled water. The table on page 64 lists the orgonotic potency (OP) values of a series of fluids. 3. Next, the sieved garden soil is boiled in distilled water or ordinary tap water for 1 hour, or autoclaved for half an hour at 120 degrees Celsius and 15 pounds pressure. 4. The water is filtered off from the boiled earth, crystal clear. We call this fluid Bion water. In contrast with the original colorless water, this fluid is one or another intensity of yellow. The fluorophotometric measurement of the orgonotic potency of the Bion water is taken immediately after boiling and filtering. The value obtained, that is, its OP, usually varies between 30 and 60 and averages about 45. In other words, the orgonotic lumination of Bion water is, on average, 45 times stronger than before boiling. Since the galvanometer records the reaction of the photoelectric cell to the fluorescent light rays, The rise in the fluorophotometric value of the water after boiling the soil indicates a higher energy content in the fluid. This can be estimated in microamperes. Understandably, however, the microampere reading taken from the galvanometer is not the real measure of organotic illumination. It is simply the measure of the excitation of the photoelectric cell transformed into electrical energy. As I have demonstrated in another context, electrical measurements record only an extremely small fraction of the actual energy values of orgone energy. 5. That boiled Bion water attains energy values approximating those of organic fluids such as bouillon, milk, or vitamins seems astounding and incomprehensible at first. But further consideration shows this result to be perfectly logical. The boiling process transforms the substance of the soil into modal, bionous matter, and energy is freed from the matter, as is unequivocally demonstrated by the internal motility, the slow rhythmic pulsations, and the contractility of the orgone energy vesicles, in contrast to the non-motility of the particles of the same soil before the boiling process. But the transformation of the earth particles into bions and the release of the energy inside the particles were not the only effects. In addition, this energy entered the water, because water attracts orgone and vice versa. The organotic potency of the water therefore rose from its own basic value to the level of biochemical fluids of high organotic potency. 6. Experience has shown that boiled Bion water develops rot bacteria even when immediately sealed airtight. For this reason, in more recent experiments Bion water has been autoclaved for 30 minutes at 15 pounds pressure and 120 degrees Celsius. This procedure at first causes the OP to drop 5 to 8 points, but it recovers its initial level over the subsequent 24 to 48 hours. The autoclaved Bion water is then refrigerated in sealed ampoules or in sterilized flasks stoppered with sterile cotton plugs. The organization of bionous and plasmatic matter from the orgone energy in Bion water, which is sterile, completely free of particles, and has a high OP, is put into several vials in test tubes 2 days after auto-elevation or boiling. The vials are sealed airtight in the test tubes stoppered with sterile cotton plugs. We divide the tubes into 3 groups. Group A is placed into a 3-layered orgone accumulator, 1 cubic foot in size. Group B is left standing in the laboratory. Group C is put into the freezer compartment of the refrigerator. As a control measure, we store similar containers of sterile ordinary water with each group. 2 to 8 days later, Group C is removed from the freezer. Before thawing begins, we are impressed by the fact that the yellow color of the Bion water has concentrated in the center of the ice in an opaque, brown-yellow spot. The surrounding ice is completely bright and clear. As soon as the ice has liquefied, whitish and brownish flakes are observed in the previously crystal clear fluid. Macroscopically, the flakes are approximately 1 to 5 millimeters in length and about L/m in width. The fluid itself is homogeneously yellow. Groups A and B develop the same flakes, but much more slowly, taking 3 to 8 weeks. Of the control groups, neither Group B, placed in the laboratory, nor Group C, frozen, develops flakes. However, control Group A, the sterile ordinary water stored in the orgone accumulator, does develop flakes over an extended period of time. Several months, but they are much less dense and more imperfectly formed than those of experimental groups A, B, and c. Microscopic examination of the flakes always shows two basic types: smooth, plasmatic, well-defined forms, in which dark granules and occasional blue bions are scattered in varying densities, or Bions heaps of Orgone energy vesicles with an intense blue glimmer and a sharp margin, cf. Figs. 43-47, Appendix. The preparations, kept sterile, show, after a few days, and especially after 2 to 3 weeks, an increase in the number of flakes. Microscopically, it is possible to establish that the flakes grow individually through an accumulation of substance and that they also divide. The thread-like, winding, serpentine, bright flakes are transformed in the course of weeks into strongly radiating Bions heaps. Culture experiment. To obtain a clearer and more demonstrable understanding of this process of growth and multiplication, some flakes are placed, under sterile conditions, in clear, sterile him water. After 1 or 2 weeks, a culture formation can be an ally observed in the samples. The flakes have become more dense. In accordance with our previous experience, the op of the cold low fluid stays at approximately the same level, or even increases. If we examine the preparations under the microscope at regular intervals, about once a week for several months, we can see, gradual biophysical changes in the flakes. In some, there appear spherical hands measuring 2-3 microns in diameter, with a margin and a strong blue glimmer. Gradually, they stretch out until they eventually assume the following bin shape. Under favorable conditions, when no premature disintegration has occurred in the preparations, these bin forms develop into contractile protozoa which move in a rapid, jerky manner. For the most part the plasma of these protozoa has a granular and striated structure, though in a few instances it is smooth, without structure. Pure cultures of protozoa can also be obtained by inoculating the supernatant fluid without disturbing the flakes which lie at the bottom. The protozoa multiply in the new culture and without difficulty from culture to culture. Observations and tests to determine the biological nature of Orgone flakes. 1. The thawed-out, flaky Bion water preparation 20 represents an unresolvable colloid, if crystal clear, yellow Bion water of high organotic potency is evaporated, a golden yellow, smooth, opaque coating forms on the bottom of the vessel. Scraped off, this coating yields a powder consisting of small, yellow to brown crystals, which we have named Orgone. These crystals cannot be dissolved again in either ordinary water or in water containing a high Orgone content. They merely swell and react precisely like flakes obtained by the melting of frozen Bion water. They, too, exhibit the phenomena of growth, multiplication, and protozoal formation. 2. Microscopic observation of organization. The flakes grow in length and width. Bion heaps accumulate more Bions from the fluid. Larger flakes sprout. Smaller ones and the small ones grow larger. The Bions frequently arrange themselves in groups. As the flakes grow larger, they become darker, showing a brown to black color. 3. Organotic attraction and sterilizing effect. The Orgone flakes react like all other Bions matter investigated so far. In the vicinity of raw bacteria, the flakes either kill the bacteria or, at least paralyze them. 4. Degeneration and putrefaction. Like all organic or living substance, The Orgone flakes degenerate by putrefaction and disintegrate into the T-bodies already familiar to us. Protozoa form more slowly and with greater difficulty in strongly degenerated preparations than in sterile ones. Degenerated preparations can, however, be freed of T-bacilli and rot bacteria again and again by repeated freezing. This seems to promote the capacity for the organization of motile protozoa. 5. Flakes that are dried and crushed into small particles burn in a flame to a black, coal-like substance, thus reacting like living protoplasm and non-living organic carbon compounds. Even if the flakes are not burned, the identical black carbon substance that has proven to be combustible in the flame develops, probably through oxidation. 6. Sugar content. Bion water with a high organotic potency tastes sweet. Burning flakes give off an odor like that of caramel. It has not yet been possible to conduct an exact biochemical analysis, but one is planned. 7. All containers that have had contact for a considerable length of time with bion water or bion earth feel fatty to the touch. 8. Bion water reacts to weakening influences like autoclavation in the same way a living organism would react, namely, with a falling off of OP, which is regained only gradually. 9. Contractility of mass-free orgone in bion water. There can be only one plausible explanation for the appearance of a concentrated, brown-yellow spot in the center of the crystal-clear ice. Mass-free orgone energy reacts in freezing water exactly as does orgone energy in a freezing living organism. It contracts and withdraws from the point where the freezing occurs. Mass-free orgone energy is therefore capable of contraction. In the process of contraction, matter is created, evidently through condensation. This process needs intensive study. 10. The orgone flakes develop into protozoa more rapidly when T-bacilli are added to the preparation. The appearance of plasmatic spindle forms of coarse structure, similar to the K3 formations, can then be observed. Cf. Chapter 6. Stages in the development of protozoa in sterile, particle-free, bion water. 1. After the bion water solution thaws, granulated plasmatic flakes of typically organic form appear. 2. Many of the individual granulae expand and develop into spherical orgone energy vesicles with an intense blue glimmer. The majority of these groups of bions merge into larger formations by association or confluence. 3. In a matter of weeks, the spherical bions become bin-shaped, but are not yet motile. 4. These bin-shaped formations then become motile in two different ways. At 3000–5000, the content of the energy vesicles reveals delicate movements of expansion and contraction. The formations stretch more and more, their membranes soften, and they begin to move about. The forms that have become markedly elongated develop serpentine, corkscrew-like movements. The accompanying sketches are drawn from life as seen with a magnification of approximately 240x. 5. When bion water fluid containing protozoa is inoculated with sterile bion water, the protozoa multiply and can be reiniculated ad infinitum. To these protozoa have given the name orgonomia. Control experiments. 1. The freezing of ordinary, non-sterile spring water or distilled water yields neither flakes nor protozoa. The organization of plasmatic matter must therefore be ascribed exclusively to the high orgone content as measured by the fluorophotometer. 2. When bion water of approximately 40–50 op is distilled, A small number of flakes can be observed immediately after cooling and a large number after actual freezing. This is additional confirmation of the preceding statement. Since seawater is extraordinarily rich in orgone, it is not surprising that it also yields flakes and protozoal growth after filtration and autoclavation. Nevertheless, there is some slight mystery here. Seawater taken at Jones Beach, on the Atlantic coast of Long Island, New York, and measured 1 hour after extraction, was only approximately 810 op. Earth Bion water with so low an op content yields no, or at best, deficient, Bionous growth, and it is therefore difficult to explain why the low op of seawater makes no difference. The phenomenon is undoubtedly important. Unfortunately, it is not possible to understand everything at once. 3. Water from autoclaved grass also yields flakes after freezing which multiply and grow. General conclusions. 1. Humus contains orgone energy. The transformation of humus in water into earth Bions causes a rise in the orgonity potency of the water from 4 to between 30 and 70, with the op of distilled water set at 1. 2. Orgone energy in high concentration colors liquids yellow. 3. Orgone energy, homogeneously distributed in a fluid at room temperature, contracts when chilled or frozen to form a brownish-yellow core in the ice. 4. Protoplasmic flakes, that is, matter, develop from concentrated mass-free orgone. 5. Fluid with a high orgonotic potency develops protozoa by way of the formation of orgone energy vesicles. 6. The formation of plasmatic matter from concentrated orgone energy indicates a general process by which matter originates. Thus, we must consider orgone energy to be the primordial cosmic energy. 7. On the basis of our findings in Experiment 20, the development of living plasma on our planet preceded the organization of coal substance and carbohydrates. Coal is a product of the disintegration of living matter. Biochemical molecules did not exist prior to the development of plasmatic substance, but appeared as one of the mechanical constituents in the process of plasmatic organization.